Welcome to the video on cell division, looking at mitosis versus meiosis and asexual versus sexual. Let's take a look. We're going to start with the cell all the way to the left. This is our sperm, and the one to the right is the egg, also known as the ovum. Inside the sperm, you have one chromosome with gene A, one chromosome with B and T. Therefore, this is a monoploid cell represented by the letter N. Same in the egg. One chromosome for A and R, one chromosome for B and T, also monoploid, mono meaning one. This arrow shows the sperm and egg coming together by a process called fertilization. And this restores the true amount of chromosomes that should be in this organism. Now you have two chromosomes for gene A and R. This is known as a diploid cell. Di meaning two, and this is represented by 2N. This fertilized egg is referred to as a zygote. This is your very first body cell, your very first somatic cell. Since you have two chromosomes with the same genes, those chromosomes are called homologous chromosomes. Chromosomes with the same genes. You find homologous chromosomes in every single body cell in your body. All somatic cells will have homologous chromosomes. You never find these, though, in gametes, as we saw. There's only one chromosome for a specific gene. So let's take a look now at what happens to that zygote. It's going to divide into two cells. You don't stay one cell big forever. And they're both going to be diploid cells, just like the zygote. Body cells divide by mitosis, and mitosis will make genetically identical cells, cells with the same DNA. Those two cells will divide into four cells by mitosis, because we're building your body. Those will have identical DNA as well. Four cells will divide into a big ball of cells by mitosis, making identical cells as well. And eventually, these cells will keep on dividing until they make you, a person, by mitosis. That kind of brings us to a little quandary. All our cells have the exact same DNA. That's why any cell could always lead back to you. But cells are able to carry out different functions and make different proteins, even though they have the same DNA, because they're going to use different parts of the DNA instructions. They're going to use different genes to carry out different functions. This is called cell differentiation. At a certain point in a person's life, they hit a stage called puberty. Once they hit puberty, if they're female, they're going to make eggs by a process called meiosis. The E stands for eggs. If they're a male, they're going to make sperm, also by meiosis. So, looking in summary, it looks like mitosis is used from one cell big all the way to you're grown. But once you hit puberty, then you're going to start to use meiosis, and only then. So before puberty, you're not using meiosis at all. So let's sum it all up. Mitosis is used for all body cells, every single one, all your somatic cells, and it creates genetically identical cells. There's no variation at all. In humans and in other organisms, it's used for growth and tissue repair. Tissue is simply just a bunch of similar cells. Meiosis, however, only makes egg and sperm cells. It only makes your gametes. So literally any cell that you say besides egg, sperm, or gamete is done by mitosis. Only eggs, sperm, and gametes are done by meiosis. And it creates unique cells that are monoploid or haploid. That word haploid has not gone over yet, but it's literally the same thing as diploid, or as monoploid. So this diploid cell has four chromosomes, two pairs of homologous chromosomes. In the sperm and egg, you only have two chromosomes each, and there's no homologous chromosomes. there's only one chromosome for a specific gene. That's why it's monoploid. But you also have half the number of chromosomes. Therefore, you could refer to the cell as haploid as well. Haploid meaning half. So monoploid and haploid mean the same thing. Looking at the cell cycle, besides the human cycle, let's start with the birth of a cell. This first section of the cell's life is referred to as the G1 or gap phase. This is when a cell grows, makes normal proteins, does what it's normally supposed to do. The second stage of its life, referred to as S phase. This is when the DNA is replicated. The S stands for synthesis, synthesizing new DNA. The final stage is referred to as the gap 2 phase, the G2. This is where all 5 billion of those letters are checked for errors, and if there's too many errors, the cell will destroy itself, called apoptosis. Then the cell moves into this area of its life, the M phase. It could be one of two types of cell division, hence the letter M. It could be mitosis, which makes genetically identical cells. Again, it's used for somatic cells in humans and animals and plants. It's used for growth and tissue repair specifically. And it's the only type of cell division in asexual reproduction, where new organisms will have completely identical offspring, no variation at all. This is really bad because if one of them dies from, say, a fungus or a bacteria, all of them will die. Quick note on asexual reproduction. Only one parent is involved, and one parent will make the identical offspring by mitosis. Here are some pictures showing asexual. This is budding. This is binary fission. Both no variation. The other direction this cell, if this is a sex cell, it could do meiosis, where 4 unique monoploid or haploid gametes, sex cells, are made. Remember, meiosis only makes sex cells. And meiosis is exclusive to sexual reproduction. It is not used at all in asexual. Sexual reproduction creates unique offspring, meaning it creates variation, and if one dies, the others may still live because they're different. They might have an adaptation, a positive trait that helps them survive better than the ones who died. Quick note on sexual reproduction. These graphics show 2 parents making a unique offspring. Taking a look at the cell cycle again, these 3 sections, the G1, S, and G2 phase, they all make up the majority of the cell's life. This is called interphase. And during interphase, the DNA is in a messy type form called chromatin. And now we're going to take a look at M phase, either meiosis or mitosis. And we're going to look at how both these processes work. All right, let's take a look at mitosis. We see in mitosis there are 2 chromosomes of gene A and B and 2 with R and T. That means this is a diploid cell. All body or somatic cells are diploid. They'll always have homologous chromosomes, 2 chromosomes that have the same genes. There are 4 total chromosomes here. Diploid cells always have an even number of chromosomes because they need to have pairs. The next stage below, this is still a diploid cell, no surprise. But the chromosomes, they're lining up in the middle, and they line up single file, straight down. This is usually metaphase, but there's another video that shows all the distinct phases, the mitosis video. And it looks like the spindle fibers are attaching to the centromere, and they form a thing called a kinetochore. Kinetochore is simply just a complex of proteins where the spindle fibers attach. That's the protein bundle right there. And that's the spindle fiber. So the spindle fibers are going to attach to the centrioles in animal cells. And ultimately, the spindle fibers are going to rip the chromosomes apart. Specifically, they're going to rip the sister chromatids, which are identical, very important, to opposite sides. So one identical sister chromatid that have like A and B is going to go to one side, and the other sister chromatid that has A and B also is going to go to the other side. So identical sister chromatids move to opposite sides. That's important because the cell on the left and the cell on the right are going to have the same exact genes. Because all the left sister chromatids went to the left side, and all the right sister chromatids went to the right side. And since sister chromatids are identical, both these cells will have identical genes. As seen down here, after telophase cytokinesis, you see these two cells. They have 2 chromosomes for each gene, and they have identical genes. This is how mitosis creates 2 cells that are identical. All right, let's take a look at meiosis. It's a type of reduction division, meaning you start with a 2N cell diploid and you reduce it down to make 4 monoploid gametes. How does this happen? To start, you have 2 chromosomes here with the same genes, A and B. 2 chromosomes with the same genes, R and T. So you have homologous chromosomes. This is a 2N or diploid cell. Total of 4 chromosomes. And just a reminder, humans have 46. You can't drill 46. These 2 chromosomes, the homologous chromosomes, it looks like they pair up. And they're forming a little group. The group of homologous chromosomes is going to be referred to as a tetrad. And in the tetrad, you could see the arms of the homologous chromosomes. They're starting to kind of cross over a little bit. They're starting to touch each other. And the point of contact between the homologous chromosomes is referred to as a chiasma or chiasma. This is it right here. So they get super cuddly, they touch, and then they literally strangle each other, the chromosomes. And that's going to cause them to create or become unique. Genes are going to be mixed or shuffled between homologous chromosomes. The sister chromatids, they're no longer identical. This is called crossing over, when homologous chromosomes physically swap genetic material, physically swap genes or alleles, versions of genes. And this is going to create new, unique genetic combinations. This is why I'm different from my brother or sister. We all came from the same gene pool, same mother and father, but we have all different genetic combinations. That's because of crossing over. It creates those unique combinations of genes. So this is still a 2N diploid cell with 4 chromosomes, but if you look in this next picture, something weird's going on. It looks like the homologous chromosomes on the left are going to the left, and the homologous chromosomes on the right are going to the right. Homologous chromosomes are splitting, and they're moving to opposite ends. This is a momentous occasion in meiosis, because ultimately, we're going to make 2 cells that only have one chromosome with genes A and B, and one chromosome with genes R and T. Therefore, the splitting of homologous chromosomes created monoploid cells. And look at the number of chromosomes. There's only 2 chromosomes in this cell now, or both these cells. Therefore, they're also known as haploid, meaning they have half the number of normal chromosomes. So, what creates, or how do you go from a diploid to a monoploid cell? When homologous chromosomes split, that will create monoploid cells. Let's look ahead a little bit. Now we have our monoploid cells. We did the reduction. What's going to split next? Because if you remember in the first image when we looked at meiosis, you go from 2N to 2 cells that are both N. And the final cells are all N. So how does that happen? I do want to mention that the equal splitting of genetic material, the equal separation of genetic material, is called disjunction. Important word to know, because if it doesn't happen, big problems could occur. So in this second division, it looks like sister chromatids split. And that's going to create or keep the chromosome number the same. All these sperm cells at the bottom, they all have 2 chromosomes. And in each one of them, they only have 1 chromosome for genes A and B and 1 chromosome for genes R and T. All of them have a unique combination of genes. Therefore, 4 unique monoploid gametes were created. And all these sperm can create new genetic combinations. Each sperm will make a brand new baby, a new unique individual. Let's take a look. Let's say this sperm hits the egg, fertilizes the egg. Let's put, we have mom's genes already there. Let's get dad's chromosomes with little a and little b, and little r and little t. This cell is a diploid cell, the fertilized egg. Let's say this sperm hits it though. Dad's going to give different genes. You have new gene combinations. So fertilization is a source of variation as well. So you could have abnormalities, problems with meiosis that could really cause some serious problems. It looks like here we have an unequal separation of genetic material. That's a problem. This is called non-disjunction. If disjunction is the equal separation, non-disjunction is the unequal separation of genetic material. And we're going to be left with 2 cells, the 2 monoploid cells. But this monoploid cell, it has its 2 chromosomes, but it has an extra one. So 2 chromosomes is the monoploid number, N, but we have 1 extra, plus 1. So this cell will be annotated as N plus 1. This cell over here is still N. It's still 2 chromosomes. Chromosomes could be single-stranded or double-stranded. Refer to the Genetic Essentials video posted very recently. And sister chromatids will divide to keep the chromosome number the same. And this cell down here is N plus 1. This cell is N. This sperm, we're missing the chromosome. So this is N minus 1. The haploid number is N, which is 2. We have 1 less, N minus 1. If this cell with an extra chromosome fertilizes the egg, it looks like the egg, when it gets fertilized and becomes a zygote, it looks like it has its homologous chromosomes, It has its 4 chromosomes, will make it diploid, but it has that extra chromosome. So it's going to be 2n plus 1. And this means you could have something like this, 3 chromosome number 21s. This will give a condition called Down syndrome, known as trisomy 21. Tri meaning 3, 21 referring to the 21st chromosome. Again, this is caused by non-disjunction, unequal separation of genetic material. You could also have this happen with sex chromosomes. You can have an extra sex chromosome. Females are XX, males are XY. And if you have XXY or XXX, you could have underdeveloped genitalia, trouble reproducing. This is a condition known as Klinefelter syndrome, also caused by non-disjunction. So here's meiosis and the 2 ways it could be performed. It can make sperm, our first sex cell. This is known as spermatogenesis, sperm creation. Or it can make an egg. And when meiosis makes an egg, it only makes 1 healthy egg. The other 3 cells are going to be referred to as polar bodies. They're going to actually donate all their cytoplasm to the 1 healthy egg. That's why they're so tiny. And the cytoplasm is going to contain a lot of rich nutrients, so once the egg gets fertilized, it could develop into a healthy embryo. This is called oogenesis, egg creation. Both spermato and oogenesis are known as gametogenesis, gamete creation. Thanks for watching. Subscribe for more.